It's my real pleasure to introduce tonight's speaker, Dr. Prashant Sharma, who's here visiting us from the University of Wisconsin-Madison. He's very interested in animal biodiversity, okay? And his focus, his favorite group of animals are the chelicerates, also known as AKA the spiders and their kin. And he basically explores biodiversity through integrating a lot of different approaches. And the first one I'll just mention because many of us, go outside. He's out there crawling around, turning over leaves, collecting different critters and basically categorizing and characterizing this biodiversity. But it doesn't stop there. He's not just a tree counter. He's very interested in making sure to understand this biodiversity in basically through time and space. So he uses very stringent phylogenetic methods to understand the evolution of this biodiversity and how these different forms are related to each other. He also has a strong interest in understanding the developmental and genetic basis of how all these different forms arise through the life cycle. So I'm not going to go further into anything. I'll leave it to him, but I just want to mention a little bit about his academic history. Dr. Sharma did his undergraduate and his undergraduate degree as well as his PhD at Harvard University. And then he received additional training at the American Museum of Natural History before taking an assistant professorship in the Department of Integrative Biology at the University of Wisconsin-Madison, where he's been ever since. So I don't want to take any more time. Prashant, we're delighted to have you. You're all in for a treat. The stage is yours.
All right, thank you so much, Elaine, and thank you so much for coming this evening. Can everybody hear me okay in the back? All right, excellent. So like a lot of the scientists here at the Whitney lab, I pursue a body of work that is very much impelled by understanding the nature of biodiversity and what it is that maintains and generates biodiversity over the course of evolutionary time. And the group of animals that I work on is broadly known as the arachnids. And when we hear arachnid, the animals that we most commonly think of are the spiders, such as the ones that are shown here. This is an immensely successful group of animals with about 54,000 described species, and the number just doesn't stop with more species described every year. What you're looking at is a group of animals that is renowned for a number of different innovations. Innovations in the uses of their silks, as exemplified by that orb-weaving spider in the top left. Innovations in their venoms, and this is one of the reasons that spiders are notorious, as well as innovations in features like sexual dimorphism, both in terms of size disparities between male and female, and also the armature, the sexually dimorphic weapons that you will find in several of these species, like the photograph that you see in the bottom left. Now, spiders are one of a number of different groups of the chelicerate arthropods. So shown here is an array of lineages that we collectively group as the chelicerates. And this is the group that includes spiders, scorpions, mites, ticks, and then a couple of marine groups like the sea spiders, the pycnogonids in the top left, and then right below them, the Xiphosaura, horseshoe crabs, animals that you are probably lucky enough to see here very frequently in Florida, in which I wish I could see more frequently in Wisconsin, but never get the chance to do so. The rest of the chelicerates are collectively known as the arachnids, and these are the terrestrial eight-legged arthropods, right? So spiders are the most commonly known amongst those. Just about everybody knows what a scorpion is, and there's an array of smaller, less well-known lineages. For example, vinegaroons, whip spiders, and one lineage of interest to me personally, the one in the bottom left image, Opilionis, commonly known as daddy-longlegs. And I'll touch on their biology towards the end of this talk. So one of the things that spiders are renowned for is the complexity of their silk. And silk is produced from a series of posterior organs that we call spinnerets. The number and arrangement of those organs varies from one lineage to the next. And beneath those appendages, beneath the spinnerets, is a staggeringly complex series of glands that produce these different silks. One thing you may not know about spiders is that different components of their webs are constructed using different silk proteins. Each of these silk proteins is associated with different glands, and the number of those glands changes dramatically across the phylogeny of spiders. So a tarantula can spin silk just like an orb weaver, but the orb weaver's web has a larger number of silks, and they're put together in a number of different complex ways. A separate feature that is of active research interest in spiders is the acuity of their vision. Some groups of spiders, such as the leucosoids, so that's wolf spiders and their kin, and then salticids, jumping spiders, are renowned for the acuity of their vision. Now, spiders usually have 8 eyes, and these differ in their number and arrangement across the families of the spiders. But one of the things that has compelled a lot of curiosity within the arachnid community is why it is that certain sets of eyes are enlarged across different lineages, and how it is that these are specialized for motion sensing, for night vision, and for, in some cases, even moving parts of the eye to track prey. One really interesting study from a couple of years ago used the movement of spider eyes during darkness to report the possibility that spiders might even dream. They might even have REM, something like REM sleep, and analogous to what we have. And that's something that for a lot of people to think about. My own contribution to the understanding of spiders and the understanding of arachnids at large lies at the intersection of their phylogeny, so how it is that they're related to each other, and their developmental biology. So among the important questions for us in that segment of the biological community, we are impelled by questions like what genetic programs make the arachnid embryo? How do you build an animal that looks like this? How do changes in those genetic programs achieve changes in phenotypes? How do you build biodiversity at the genetic level? How do you go from an archetypal spider like the one at the top left to something like an ant-mimicking spider in the photograph right below it, to some of these really bizarre lineages that are camouflaged to look like pieces of plant matter? And then beyond spiders, among the things that we concern ourselves with is how do novel traits come about across arthropod diversity? Now, we've talked previously about things like spinnerets and eyes, but outside of spiders, there's a staggering array of novel traits that are lineage restricted. You only find them in one particular branch of the tree and nowhere else. And these include, for example, the pectines in the top left of scorpions. These are gill-like external mechano and chemoreceptors that only scorpions have. Or even the case of appendages that various lineages bear, some of these are specialized in incredible ways to serve various functions. For example, the palps that spiders will often use for sensing and courtship are modified into these really bright red raptorial spines in a group of animals called the whip spiders, not true spiders. These lineages use those same structures for prey capture instead. How is it that those changes are achieved? And the focus of my research group very much comes from the context of the evolution of spider genomes. About 9 years ago, one interesting discovery that we stumbled into is that the common ancestor of spiders and scorpions shares a phenomenon called a whole genome duplication. This is an ancient event that resulted in the birth and the retention of 2 copies of lots of genes that occur as single copy in groups like insects or centipedes, right? Now, spiders, scorpions, and a handful of other lineages in that part of the arachnid tree have a whole genome duplication, but other arachnid orders, such as mites, ticks, daddy-longlegs, and purple, do not, right? Horseshoe crabs have their own set of whole genome duplications, at least 3 of them, but these have nothing to do with the ones that occur in the arachnids, and those are of interest to horseshoe crab researchers for very separate reasons. And the way that we approach these questions, to some degree, the way that this was discovered, part of it had to do with genome sequencing, and the other part of it had to do with the examination of embryos. Shown here is a series of gene expression patterns, the surveying of gene expression domains of genes in 2 different species. Underneath the red bar, you have a daddy-longlegs. This is one of those lineages that does not have a whole genome duplication. And on the right-hand side, you have spiders. Were we to zoom in on one of these, for example, what you'll notice is that the daddy-longlegs in red has one gene that we call MSX, spiders have 3 copies. And what you'll notice here that's of really interest to us is that each of those 3 copies has slightly different expression patterns from another. So MSX1, 2, and 3 do slightly different things. And this appears to be the case throughout the genes that are duplicated and faithfully retained in spider genomes. Could it be that this is part of why spiders are successful? Could this be what builds spider traits? This is really what has been compelling arachnid developmental biologists over the course of the last decade. So in the course of the next few minutes, what I'll take you through is our investigations into what some of these new genes might be doing and whether these successfully explain something about spider development and diversity or whether they do not. I'll tell you a little bit about the evolution of segmentation as understood through the lens of spiders, and I'll tell you a little bit about visual systems as well. And jumping right into segmentation, the nature of arthropods, being a member, every single member of this phylum happens to have a body that is composed of repeated units called segments. Right? Every single arthropod is composed of segments. And groups of these segments are frequently fused together to serve coordinated functions. We refer to those units as tagmata. So in an insect, the body is divided into 3 different units: head, thorax, and abdomen. Now what you'll notice here is that if we compare the insect, this cricket, for example, to a spider, it's organized in very different ways. And whereas for example, all the locomotory appendages, all the legs of an insect are found only in the thorax, in a spider, the feeding, sensory, and all of the locomotory appendages all occur at the anterior unit, sometimes called the cephalothorax, more formally known as the prosoma. And it's got a back end, which we call by a different name. It's basically an abdomen. We call it an opistosoma so as not to confuse it with the insect counterpart. So the front end of a spider is called a prosoma, and the back end is called an opistosoma. They're connected by this very narrow tube of tissue that is called a pedicel. And despite a fair amount of understanding of how segments are built, the paradigm that really emerged over the last, I want to say 25 years or so of investigation into segmentation is that segmentation is by and large achieved through a series of coordinated gene signaling networks. A gene regulatory network establishes segmentation in one of the best studied arthropod models, that's the fruit fly, Drosophila melanogaster. And as insect biologists compared the activity of those genes across insects, they found that, well, these genes appear to do very similar things, give or take. There's some variation here or there. But if we compare the activity of these genes in a fruit fly on the left, Manduca in the middle, and Tribolium is a beetle on the right, so insects with different modes of segmentation, we nevertheless find that a lot of the same genes are recycled and used in very similar ways to build these repeating units. And the paradigm that emerged from this was that it appears that arthropod segmentation is something that was present in a common ancestor. It was built using a concerted set of genes that must have been present in the arthropod common ancestor, and the segmentation program is faithfully conserved across these lineages. So far, so good. The challenge here, of course, is that we happen to be looking at a fairly small branch of the arthropod tree of life. Much of this textbook understanding, this paradigm, was informed by insects. We focus on chelicerates in large part because they sit at the very bottom of the arthropod tree of life. So they're the sister group to all of the remaining arthropods. So trying to get an understanding of how it is that particular processes emerge and evolve, you have to consult something like a spider or a horseshoe crab. You have to have comparative data spanning the opposite side of the tree. Similarly to body segments, the legs of arthropods and the namesake, what gives the phylum Arthropoda its name, is the jointed leg. Right? So just like the body parts, the legs of arthropods are similarly composed of segments. And these are formed for example, in a fruit fly, these are formed at the posterior end of a segment, and a suite of genes is required for activating that leg program. So at the back end of a segment, 2 genes called wingless and engrailed establish the identity of that posterior region. Wingless, WG in this figure, activates another gene downstream called SP69. sp69 will wake up distalis, and these two genes together, sp69 and distalis, are required for leg fate. If we create a mutant that lacks distalis altogether, you end up with, well, a fruit fly that has a distalless leg. The entire distal territory is deleted, right? If you create a mutant for sp69, a similar thing happens. You delete that entire region. And one of the things that's interesting to us, arthropod biologists, is that even though these legs are composed, as an insect leg versus a spider leg, even though they're composed of different numbers of segments, it appears that at least some of the genes are doing something very similar. They appear to be expressed in similar ways. But at the time, we didn't quite know very much about how some of these genes work. That is, we knew how they were expressed, but we didn't know if they did the same things. So several years ago, one of the things that my lab pursued was an investigation of some of these leg patterning genes. And this work was led by a former graduate student in my lab. She began this work as an undergraduate. Emily Sutton, actually a member of my host Elaine Seaver's lab, who has unfortunately left the world of arthropods to work on annelids, a fact about which I'm not bitter at all. And it's okay though, because annelids are also segmented, so that does make it okay. One of the things that Emily aimed to do was disrupt this gene sp69 and then also distalis in a spider. She wanted to see what happens when you knock down that gene. And the expectation is that when you knock it down, the distal part of a spider leg should disappear. Straightforward. But when she did that experiment, and here you're seeing an embryonic spider, this is what it looks like just before it hatches, kind of looks like a very tiny baby spider. This is in lateral view. When you knock down sp69, a funny thing happens. Yes, you delete most of the distal parts of the appendages. You lose the distalmost segments of the legs. But two segments of the body, what bears leg 1 and 2, also disappear. And Emily aimed to understand why this is the case. What she found is that in contrast to insects, this gene sp69 is expressed very early in those two leg-bearing segments. It's picked up an additional role in patterning entire body regions. She then performed a series of experiments interfering with these genes early and later in development. What she found is that both sp69 and distalis, these leg patterning genes, appear to have the same function. So here's your normal spider on the left. If you silence distalis early in development, so the gene is shut off temporarily and then it will eventually turn back on. Well, in that case, you will elicit an early function, an early loss of function effect, where the first and second walking leg segments of the head are deleted. So you end up with a four-legged spider. Delete it later, if you knock down the gene later, you end up with a canonical classic phenotype that we know from the insect literature. All the legs go away. And knock down sp69, you end up with a blend of the two. You lose both the first two walking leg segments, and you lose the distal parts of all the legs. So what Emily has essentially discovered then is that spiders are making head segments using leg patterning genes. And this was a really surprising outcome. For a long time, we had thought that the genes that make different body regions of spiders were the same as the ones that are required for making those body regions of things like fruit flies. But one of the gaps that emerged in the literature is that as you start to explore those networks, there's actually surprisingly few genes that are conserved across insects and arachnids. Arachnids appear to be using different genes altogether to be making those body regions. And what Emily has found here is that, at least in arachnids, they have done something interesting with their leg patterning genes toward making body segments, and it appears to fill a gap in the literature in explaining how it is that these genes are recruited to make those body regions. So this suggests then that we've missed something by ignoring spiders up until now when we explored the evolution of segmentation. They're using ancient genes, but they're using them in very unexpected contexts. Now, one of the ways that we aim to accelerate our understanding of what are the genes that build different parts of spiders, one of the ways that we can increase the rate of their discovery is that rather than pull genes from the literature, candidates from the fruit fly literature, for example, we can look at it by examining what genes are expressed in different parts of the embryo as it develops. And the way that we'll often do this with different species and different orders of arachnids is that we will take stages of embryos, we will excise, cut out different body segments, and then we will sequence them. And we do this across several different stages of arachnid embryos. So those regions of the body are extracted, we generate short read sequencing libraries, and then we map those reads back to the genome. It is essentially a way of asking the elementary question: what's highly expressed in this region of the body, and what's lowly expressed in this region of the body? What important genes might be active in different parts of this axis? After mapping those reads back to the genome, we compare them bioinformatically, right? So we use the distribution of the reads as a proxy for how highly or lowly a gene is expressed. And to approach this question in spiders, Emily established a number of resources for the largest spider embryo she could find in North America. It was the embryo of a tarantula, very easy to dissect because this thing is about 4 millimeters in diameter. And she excised a number of different body segments, starting from the anterior all the way to the posterior. So she pulled out regions of, for example, like the chelicera, so this is the fang of a spider, the palp, the sensory appendage, legs, and then as well as posterior body regions, like the spinnerets and then the book lungs, the respiratory organs. What she was able to show through that approach is that a lot of the anterior organs, so legs and palps, for example, and the fangs, they all happen to be expressing genes somewhat similarly. This is shown as a principal component analysis. So the proximity of those colored dots that correspond to those different appendages on the left, it tells you how similar the expression profiles of those libraries are. So it looks like all the anterior segments are expressing very similar genes, and in all the posterior regions, for example, the book lungs and the spinnerets, interestingly enough, the web spinning organs and the respiratory organs start out expressing almost identical complements of genes. They're essentially indistinguishable early on in development. What it is that changes them into a web spinning organ versus a respiratory organ happens later, and that's something we're still working on understanding. But broadly, when this process is complete, once you identify the distribution of genes across this axis, what you aim to do is ask what's highly expressed in one region of the body versus another. In this heat map, all the rows that you're seeing, each one of those rows is an individual gene, right? All of our different libraries are in the top. So those are replicates or 3 replicates each for every one of those different tissue types. And using this kind of approach, by examining thousands or tens of thousands of genes at a time, it becomes much more straightforward to extract bioinformatically. For example, let's find all the genes that are really highly expressed in the opistosoma, in the abdomen-like territory, versus genes that are really lowly expressed in the prosoma. So what's important for the abdominal region? What makes that part of the body? And through this approach, Emily was able to identify a number of different candidate genes, and she pulled out something like 16 and silenced 16 of them in separate experiments. 14 out of the 16 didn't seem to have any effect, suggesting that maybe they don't do anything as important as we imagine, but 2 of them did result in a phenotype, and one of them is particularly interesting. That particular gene belongs to a family of transcription factors that we call the Iroquois complex. Iroquois complex genes do all kinds of important things, including in human development. In this case, she happened to find a copy of a gene called Iroquois 3, which has a very interesting evolutionary history. Iroquois 3 is shown here, expressed in a spider embryo. This is a much younger stage of spider embryo. So when they develop this early, the reddish part is the yolk, and the blue counterstained part, this is the embryonic tissue. So this is the developing spider, so they're absolutely adorable at this stage. They start as a C shape, right? And what you'll notice is that after the last leg ends, after the 4th leg, L4 is when you start to see stronger expression of this gene. Now, I told you this gene has a really fascinating evolutionary history. As it so happens, we don't know anything about the function of this gene. And that might surprise you, because a number of people, a very large number of people, have spent decades working on the functions of lots of genes in the genome of the fruit fly, Drosophila melanogaster. We know enormous amounts about how it is that different genes function in the fruit fly. And as it turns out, nothing in the fruit fly can tell you anything about the origin of this gene or its history. And here's why. Shown here is a gene tree for the Iroquois complex, right? There are 3 copies. Those 3 copies appear to have been born in the common ancestor of the arthropods. So outside of the arthropods, for example, if we look at a velvet worm and a tardigrade, they only have 1 copy of the Iroquois genes. But what's interesting about this orange copy, about Iroquois 3, is that it does not occur in hexapods or crustaceans. You'll find it in myriapods, you'll find it in centipedes and millipedes, and you'll find it in chelicerates, but you will not find it in any living crustacean or any living insect. This is to say that this gene appears to have been lost altogether in the common ancestor of pancrustaceans, so crustaceans and insects. This is to say that we didn't know this gene existed until we looked in something like a spider or a centipede. And this is why sampling genomes broadly across biodiversity matters, not just for understanding the origins and the death of gene copies, but also for discovering new functions. So I told you before that spiders are part of a group that's undergone a whole genome duplication, right? So that group, that clade, that lineage of arachnids that's undergone a whole genome duplication, it's got 2 copies most of the time of all of these genes, pink, green, and orange. One of those 2 orange copies is the one that we're going to be interested in. Iroquois 3-2 is what Emily calls it. And so Emily aims to understand its function, so she knocks it down, she silences it in developing spider embryos. So shown here are 2 different stages of developing spider embryos. So that's a young stage here in panel A, and it's a slightly older stage in panel B. When you knock down Iroquois 3-2, this new gene copy, when you knock it down in a spider, a really funny thing happens. You end up obliterating the middle region of the body. You end up with an embryo that is missing the posterior legs, part of the anterior abdomen, and the entire middle region is completely gone. You've just obliterated the middle section. And this is something that Emily proceeded to confirm using gene expression patterns. So she used well-known marker genes. For example, a ventral midline gene called SOG, short gastrulation. And what she found is that when you examine these embryos that are knocked down for this gene copy of Iroquois 3, you end up truncating and deleting this middle territory altogether. So it suggests we found a really important segmentation gene that was present in the common ancestor of the arthropods, but is lost altogether in the common ancestor of crustaceans and hexapods. What Emily has essentially found then is a new segmentation gene. She ended up calling that gene by a new name. She ended up calling it wasteless, right? As in the gene that the disruption of which causes the loss of the spider waist. And this was one of the very few studies to date that has been able to link the origin of a new spider gene copy with some sort of function. We know of something like 1,000 genes in the spider genome that are duplicated since the whole genome duplication. We know the expression patterns of lots of them, but we know very little about their functions. And as I said, between then and now, I think there's something like 3 or 4 studies that have shown that these new spider genes are doing something new. So this then suggests to us that there may indeed be new genes that are associated with new functions that result from the whole genome duplication. But it's important that I stress that there are some things that those new genes can explain. They can't explain everything, and here's what I mean by this. So far I've shown you how it is that new genes can establish new territories in spiders and possibly other arachnids as well. Let's look at an older character system. Let's look at visual systems. So I told you before that spiders, or at least some groups of spiders, are renowned for visual acuity, right? Now spiders typically have 8 eyes, and these are divided into 2 visual systems. One pair of eyes, one of these is false colored here in yellow, is called the median eyes, right, or the principal eyes. And the remaining 3 pairs are collectively referred to as the lateral eyes, right, or the secondary eyes. You'll notice here that primary and secondary doesn't really correlate with size, for example, but these eyes do differ markedly in structure. Shown here on the left is the anatomy of the median eye, and on the right is the anatomy of the lateral eye. Right? And one of the things that differentiates the two is the presence of this blue, what's labeled in blue, a crystalline, a reflective layer called a tapetum. The tapetum is what gives spiders their eye shine. If you ever go out at night and wave a flashlight around and you see a pair of bright dots reflecting back at you, follow it to its source and you'll find a spider there. That's the tapetum reflecting at you. So these eyes differ in the orientation of their photoreceptors as well, and how it is that they're arranged relative, with respect to the other eye parts, with respect to the photoreceptor bodies. They differ markedly across lineages. They differ markedly in exactly what they can do. Sometimes those median eyes are image forming. Sometimes the lateral eyes are used only as motion sensors. And they, as I said before, they vary markedly in their arrangement across different spider lineages. Now, what's important to know is that these two visual systems are ancient. They appear to have originated at least as far back as the split across all the arthropods. And the reason for this, what you'll notice here, is that the eyes differ markedly between spiders and something like an insect, like a fruit fly. Right? So fruit flies typically have this very large and obvious pair of faceted eyes, compound eyes. And at the very top of the fruit fly's head, what you'll notice here is a trio of very small reddish structures, right? And those are ocelli. As it turns out, the ocelli, which I've labeled here in blue, those are the homologs of the median eyes, the evolutionary descendants of the same program. By contrast, the lateral eyes of spiders are thought to be the disaggregated remnants of the compound eyes. Right? This is inferred both from the fossil records, so there are fossil arachnids that have these different stages of compound eyes that become less and less aggregated. It's also inferred from where in the embryo these eyes originate. So the lateral eyes, as the name suggests, start out on the lateral edges of the head lobes, and the median eyes occur more anterodorsally. The other aspect of arthropod visual system evolution that's fascinating is that we've often focused on spider vision, both as a result of their visual acuity, but also because they retain both of these visual systems, the median and the lateral eyes. Not all arthropods do this. There are various arthropod orders that have lost one or both pairs, or one or both sets of eyes altogether, especially miniaturized lineages like mites. But it is thought that, and one of the reasons we really focus on spider vision and spider visual system development is because it is thought that we see traces of those eyes in some living lineages. And we see additional pairs of eyes as well as rudiments or vestiges. So were we to look at a developing horseshoe crab as an example, I told you before, this is another group of chelicerates. Horseshoe crabs do indeed have compound eyes, so they retain faceted lateral eyes. They also have a pair of median eyes that occur more anteriorly. Those are seen as a pair of black dots at the very anterior end of this trilobite larva. And what's more, they'll also have additional larval eyes that are much harder to detect. One of them is a rudimentary lateral eye that occurs in the posterior end of the compound eye, and this is something that is retained, at least as a rudiment, through most throughout its post-embryonic development. And then more anteriorly to those lateral eyes, there'll also be a third median eye. It starts out as a pair that ends up becoming fused. Right? There are additional eyes that also occur in horseshoe crabs, like the paraventral eyes and parietal eyes and so forth. But what I'm trying to get at is that visual system evolution is fairly complex across the chelicerates. It does not help that, as I said before, various lineages have lost one or both pairs of eyes. For example, in sea spiders, the sister groups of all the other chelicerates, they have 4 eyes, but the 4 eyes are all median eyes. So they have 2 pairs of median eyes. They essentially retain what you might find in a very early stage of a horseshoe crab, but they've lost the faceted eyes altogether. So you can probably guess why it is that people around the world, and particularly in places that are far away from the shore, have focused on spiders as the model, the leading model for the evolution of vision. They're accessible, they're tractable, and they have both median and lateral eyes. It's kind of, as I said before, I'd love to work on horseshoe crabs, but it's kind of hard to do when you're in Wisconsin. So one of the directions in which people have focused this investigation is to look at a particular cascade of genes that we know is important for patterning eyes across bilaterally symmetrical animals. And we refer to that family of mostly transcription factors as the retinal determination gene network, or the RDGN. Some of these genes you might have heard about before. The name PAX6, for example, might ring a bell. These are essential genes for the establishment of eye fate. So some of these classic experiments from the fruit fly literature from the 1980s and 1990s were able to show that misexpression of some of these genes, like the PAX genes, results in ectopic or misplaced eyes in different parts of the body. So if you overexpress a PAX6 gene throughout the body of a fruit fly, for example, all those additional reddish patches you're seeing at the tips of the antennae, for example, or on the wings, or on the legs, all of those are ectopically developing compound eyes. So these genes are essential for establishing eye fate. By themselves, they're sufficient to induce eye development. They're connected together in a very complex network that, as I said before, we'll abbreviate as RDGN, the retinal determination gene network. Now, spiders, as I said, have both visual systems, and we know that in their genomes they have homologs, evolutionary descendants of all of these genes. So that makes it a really useful point of comparison. But one hang-up, again, is that whole genome duplication. As it turns out, they happen to have 2 copies of almost all of the RDGN genes, not just the ones that are shown on this page, but also additional genes that are part of the network. And that creates an additional layer of complexity. We do know that a lot of the copies of these genes are transcriptionally active, and they appear to have diverged in their spatial and temporal expression patterns. As an example, shown here is, again, a spider embryo, a fairly old embryo, and this is from a study published by a couple of colleagues from about 10 years ago, right? And what you're seeing here with the dark shading is the expression of one particular gene called Cineoculus-1. Cineoculus is essential for eye development in the fruit fly. Mutants for Cineoculus will lose their eyes altogether. The arrowheads are showing you the different sets of eyes. So the median eyes are here, the black arrowhead, and the lateral eyes in the white arrowhead. Notice here that Cineoculus-1 is expressed in all of the eyes. Cineoculus-2, on the other hand, is only expressed in one of the 3 pairs of the lateral eyes. It's only the anterior lateral eye. Let's simplify that. We'll show that as this series of circles. All right? So the large circles in these icons are going to be our median eyes. The small circles will be our lateral eyes. And then a filled circle for a particular color means that a gene is being expressed there. Right? So here what you'll notice then is that there is a difference in the expression pattern between Cineoculus-1 and Cineoculus-2. And what a number of colleagues have done around the world is they've examined the expression patterns of lots of these genes in different lineages of spiders with different degrees of visual acuity. Right? So this species, for example, Parasteatota tepidoriorum, the common house spider, it's not really an active hunter, right? It's an ambusher. It sits in its web, it sits very passively, and it waits for flies to come into its web. By contrast, were we to look at a Cupienia salia, so this is a tricholaid. This is a relative of wandering spiders, a very large Central American species, active hunter. It does not sit around its web, it goes and chases prey, right? So it's a visual predator. It has a very different size of eye, and the eye has a completely different degree of capability. What you'll notice here is that the expression patterns of copies of these genes vary markedly from one lineage to the next, and they also vary between the 2 copies of these 2 different sets of spiders. And this is just a subset of the data that are available. Other colleagues in the UK have examined other spiders still, and it turns out there's quite a lot of variability in how these genes are being expressed. So the working model is, could it be that these duplicated genes acquired these new functions and regionalized different sets of eyes and thereby created eyes that are capable of different degrees of visual acuity. This is the working model. This is our understanding of how it is that you make such diversity of eye types, arrangements, and capacities across the spider tree of life. The hang-up is that there's no functional data here, or at least there weren't, right? We didn't really, a lot of this was based upon expression patterns, but we didn't really know what most of these gene copies do. And that's a problem, because another competing hypothesis is drift. That is, there's just functional redundancy here. These expression patterns just evolve like everything else does, and maybe they don't have that much functional significance at all. Maybe they do nothing, and we didn't know. So one of the things that my lab did a few years ago was we went after this question, and we started with the most obvious candidates. We took cineoculus 1, or cineoculus A. That's the one that's expressed in all of the eyes, right? Then we took two particular copies, orthodontical B, which is only expressed in the median eyes, and then optics B, which is only expressed in the lateral eyes. And we knocked down all of them. The expectation here is that if we knock down cineoculus A, we should lose all the eyes. And if we knock down orthodontical B, we should only lose the median eyes. Knock down optics B, lose only the lateral eyes. That would make a lot of sense. We would love the elegance and simplicity of such a combinatorial model. And one of the nice things about spiders is that they continue to develop their eyes fairly late in development. So the way that we watch their eyes develop is that we'll stick them to a piece of tape when they're hatchlings, and then we'll let them grow. You'll notice here that this animal's eyes are darkening as this time lapse progresses. Those dark smiley shapes that you see smile-shaped eyes that you see underneath that become very bright when the animal hatches, those are the median eyes, and the lighter circles are the lateral eyes. And were we to flat mount the head of this embryo this hatchling just before it completes that molt into the first instar, you would see something that looks like this. So again, that's the median eyes in the middle, and then those moose track shaped structures that you see there, what divides all those lateral eyes down the middle, that's the tapetum. So that's the canoe-type tapetum. When we silenced cineoculus A, for example, a very interesting thing happens. You get a spectrum of phenotypes, right? So you get a spectrum of phenotypes where sometimes it's only one median eye is affected, sometimes both median eyes are affected, but the lateral eyes are fine. And in the most extreme cases, you lose almost all the eyes. But it turns out that sure enough, cineoculus 1 or cineoculus A is required for all the eye types. And we got really excited about this because this was very promising. It turns out that the expression of this gene exactly correlates with its function. And the problem, though, and this is the part where we get really upset at the challenges of science, is that when you knock down the other pair of genes, optics B or orthodontical B, there's no result. There's no outcome. And we can measure the knockdown. It's very clear to us that we can have on-target knockdown of these genes. But despite this, even though you reduce the expression levels, there's no physical result, right? And as it turns out, since that work, we've knocked down all of the copies of these genes that are expressed in different ways between eyes and between different species. And individually, almost none of them have very much to do with eye patterning. So yes, we got an eye phenotype from cineoculus 1. You can make blind spiders, right? But if you knock down orthodontical 1, you get a headless phenotype. So you create a spider that has no head, which is always entertaining to look at. More segmentation phenotypes. And lastly, another group of colleagues has shown that if you knock down dachshund 2, there's no eye phenotype. You get a leg-related phenotype, but most of these have nothing to do with eye patterning. Now, why might that be the case? Why do we hit a brick wall here? One of the complexities of whole genome duplication is that it often results in something called genetic compensation, where one gene copy can rescue the effect of another. So even though you're silencing the function of the one, its backup copy essentially increases its expression level and can substitute that function. So there is a redundancy built into the system that we might not be able to circumvent. So it occurred to us that rather than focus then on spiders, right? A group of animals that has these duplicated genes, why not simplify the problem? Why not examine a group of arachnids that has not undergone a whole genome duplication? Now, that's a little bit challenging to do because most of the leading models in that regard tend to lack both visual systems, right? So ticks, for example, there's all kinds of advanced functional tools in ticks, but they don't have eyes, right? At least the leading model of them doesn't. So the animal we focused on was one that the lab essentially created as a model system a few years ago, and that's in the daddy-longlegs, the Opiliones. Commonly mistaken for spiders, this is a group of animals that does not spin silk, it does not have venom, right? And crash course about the systematics, the classification of daddy-longlegs, there's two groups. One of them is called the cyphophthalmi. Think of them as daddy short legs, right? And everything else that we think of when we think daddy longlegs, harvestmen, you know, like that whole group, we call that phalangida, right? The two groups both have exactly one pair of eyes, but which pair of eyes it is different between the two. Mite harvestmen, the cyphophthalmi at the top, are thought to have one pair of lateral eyes, okay? And so that's the whitish structure that you see on the side of the head. By contrast, in Phalangida, the thing we think of when we think daddy longlegs, the eyes, it's the little black dot that sits on that periscope like turret that you see on top of the head. And so they appear to have a pair of median eyes. So one group of harvestmen has only lateral eyes, one group of harvestmen has only median eyes, right? Now, why is this an interesting system to look at if they appear to only have one visual system each? Well, the reason it's of interest to us is that a few years ago, we encountered a fossil. A paleontologist colleague of mine, Russell Garb, and I described this back in 2014 from this Carboniferous nodule. This animal is about 305 billion years old. And what you're looking at is a three-dimensional scan of the inside of the space that's left inside of this nodule. And what we observe in this particular fossil is traces of both median eyes and lateral eyes, right? So it has the little promontories that we associate both with that median eye, so that little periscope-like turret that we call the ocularium, and then it's also got the lateral eye emplacements that you would find in something like a mite harvestman. So we ran a phylogenetic analysis, and what we recovered is that this lineage, which we call tetrophthalmi, it happens to fall in between the two, right? So the thinking here is that ancestrally, daddy longlegs must have had both eye types, median and lateral. And one lineage kept on the median, and the other one kept on the lateral. So to investigate how it is that daddy longlegs lost their eyes, and more generally how median eyes are patterned, we began working on this species here. So this is Phalangium opilio. This is a very common species of daddy longlegs that originated in Asia, and now it's just about everywhere in the northern hemisphere. This is one you probably see outside when you're looking around in your garden. And this system has been worked on by a number of different people in the lab. And over time, things that we've been able to develop for it include tools for surveying gene expression, genomic resources, and then tools for gene silencing, for testing the functions of genes. And we began by asking a very simple question, like how do you make median eyes of daddy longlegs? So let's just look at the single copy genes of this animal. No more two copy problems. We don't have functional redundancy. One copy each, just like insects. Let's go see what some of those do. And when we examine the expression patterns of a couple of these genes that are important for eye patterning, we notice something weird. Those very large lobes at the very anterior of the head, here you're looking at the embryo and the embryo is looking straight at you, those very large lobes that you see are what will become the median eyes, the true eyes of this daddy longlegs. But were you to look on the side of the head, what you'll notice with these arrows is that there's this little territory off on the anterior margin of the carapace that also expresses both of these genes. Why is that important? If you were to take those two genes' proteins and you shot them up pretty much anywhere in a fruit fly, it would create an ectopic eye, right? So what are these two genes doing, hanging out together in this anterior margin of this body part? And this work, which was led by another former student, Guilherme Gainet, it investigated this process. Could this be the remnant of the lateral eye, for example? And so the way that he went at it was to examine the expression of a family of genes called the opsins, which you may have heard about as well. Opsins are a class of genes that are absolutely essential for the detection of light across bilaterally symmetrical animals as well. And we know that different sets of opsins are expressed in different types of arachnid eyes. So for example, in a spider embryo, we will find one particular opsin called paropsin in all 8 of the eyes. In a scorpion, we know, again, there's copies of these genes, but we know that one of them, one R opsin, for example, is in the median eye, and then that purple gene and another sister copy, R opsin B, are both in the lateral eyes. So those are the accessory eyes, the eyes on the side of the head of a scorpion. And so Guilherme sought to understand what some of these opsins do in a daddy-longlegs. And when he did this, a funny thing happened. He was supposed to find opsins expressed only in the median eyes. What he found instead was this. The purple shading is showing you the median eyes. The green shading is showing you the expression of another visual opsin, right? The R, and it's an R opsin, RH1. And oddly enough, this opsin is expressed both in the lateral territory. It's very brightly expressed in the lateral territory. And there's also a very small group of cells, a paired group of cells in the middle of the head, just below the median eyes as well. If you knew nothing else about this animal, you would look at this and say, oh, it looks like it has 3 pairs of eyes. There's a true pair of median eyes in purple, there's vestigial median eyes in the small cells in green in the middle, and there's vestigial lateral eyes as well. And this became our working hypothesis when we saw that these lateral territories are both expressing opsins and they're expressing important genes that make eyes. Now, one hang-up, of course, that you might have is, okay, this is all great stuff, but maybe this is just an embryonic vestige, right? Lots of embryos will develop and they'll have, these rudiments that will disappear by adulthood. Are these actually important in any way, or are you just looking at sort of the shadow of ancestry? Maybe this is just kind of what horseshoe crabs had, and now a couple of these arachnids express them as embryos. Well, the test is we examine expression of a lot of these different genes through stages of development. And so were we to examine a hatchling of a daddy-longlegs, oddly enough, you would find that R opsin, that visual opsin, still being expressed in the lateral margins of the head. Were you to examine a freshly molted adult, right? So here's an adult in lateral view. That bright region at the very top is the eye, the median eye, the true eye. And this very small region on the side of the head that I'll magnify here, it expresses visual structural proteins as well as a UV opsin, so an opsin that's important for the detection of day versus light. Day versus night, right? So this is an eye-like, or at least a photosensory-like structure that appears to be retained all the way through adulthood. And this was staggering to us because for the last 100 years, the literature has always held that daddy-longlegs have 1 pair of eyes. There was a time when you would look that up and it was in your Wikipedia article, right? So we were quite surprised by this, and we really went at this further to understand, are these really eyes, or is this just some other oddball structure? And one of the ways that we went at this was to look at the neuroanatomy of daddy-longlegs. So in this brightly colored photo, you're looking at the underside of the brain of a daddy-longlegs, right? And it happens to have a couple of regions that are conserved across the chelicerates, like this arcuate body down here. These 2 lobes that you're seeing here, the MC and the LC, what we call median center and lateral center, they happen to be the regions that are connected to the median eyes and those vestigial lateral eyes respectively, right? And this was something that we examined by fluorescent labeling and by tracing these neurons. So these vestigial, what we call the vestigial median eyes, just like the true eyes, are hooked up to the MC, the medial center. And then if we trace the lateral eyes, it turns out these trace back to the lateral center. And this was something that we examined through a series of scans and a number of fluorescent labeling experiments. Why is that important? The reason this matters is that if this were a spider or a scorpion, in the same way that we see median eyes connected to a median center, lateral eyes connected to a lateral center, you would see that very same arrangement in something like a spider or a scorpion, an animal that had both sets of eyes. So this is exactly consistent with our designation of these sets of eyes as median and lateral eyes. What's more, we found that these eyes similarly respond to genetic experiments the same way. So if these are indeed true eyes, we would expect that knocking down genes that make eyes should make them go away, right? So if we knock down one particular gene called eyes absent, for example, here's the normal-looking daddy-longlegs on the left. Knock down that gene and the eyes go away. We then examine the effect of knocking down this gene on the vestigial median, vestigial lateral eyes, and sure enough, those will go away as well. You can absolutely delete all 3 pairs of eyes by knocking down the gene eyes absent. This is consistent with our interpretation that these are indeed under control of the same genetic program. And all of this is to say that we began this series of experiments by asking, through molecular markers, through neuroanatomy, and through functional genetics, how is it that daddy-longlegs lost a pair of eyes? What we actually found instead is that daddy-longlegs actually retain lateral eyes and possibly vestigial median eyes as well. And what this suggests then is that the brain architecture and the visual system of daddy-longlegs is actually more what we call plesiomorphic, more ancestral than a spider or a scorpion, which very much changes the way that we approach the origin of spider vision. A lot of the things that we thought were spider-specific or present only in this special derived group of arachnids, it appears to be something much older. There's one last coda to that story, right? And that has to do with our understanding of evolution itself. I told you before that we used to believe that there was a 4-eyed fossil once, and that each of 2 different living lineages of daddy-longlegs had lost 1 pair of eyes. Now, knowing that vestigial lateral eyes still occur in modern daddy-longlegs, what does that do to our understanding of the fossils? And to investigate that question, we performed again a phylogenetic analysis that we had run years ago. Right? So this is a crash course on daddy-longlegs relationships. There's your mite harvestman in red, and then each of these 3 colors at the bottom, these are suborders of daddy-longlegs. Phylogeny, the animal I've been telling you about, lives in this orange group down here. The fossils that I was telling you about, those are in that purple shaded box in the middle. Right? So we thought that they went somewhere in the middle of the tree. Our first question was, is phalangium special? The fact that it has these vestigial median and lateral eyes, is that something unique to phalangium, or could these lateral eyes occur in other groups? And the animal that we focused on was one lineage inside of this blue clade here. And the selection of that lineage has to do with the representation of the split at the base of phalangida. We need somebody from blue and somebody from orange to span that basal split. Fast forward through a whole lot of methods development on this Brazilian species. This is a really cool group of daddy-longlegs from the tropics. Fun fact, they're very dedicated fathers. What you're seeing is a male guarding a group of eggs and also hatchlings on a leaf. We don't know why it is, it's always the males that do this, but it's the males. And that behavior has evolved like 15 times independently, only in this group of daddy-longlegs. But we've managed to get embryos of these animals, and sure enough, it turns out they appear to express opsins on the sides of their head as well, kind of like where you would see the lateral eyes of Phalangium apilio or of scorpions. So now we rerun our phylogenetic analysis. We're just going to make 2 quick changes. The first change we're going to make is that we're going to code this time Phalangium and this species, Iporangiae, we're going to code them as having vestigial, or we will code them as having lateral eyes or present. We used to think they were absent, now we're going to say they're present. And the second change we're going to make is, for everything else, we're going to put in a question mark. We don't know whether you have lateral eyes or not. That's a fair statement of our understanding of things. And then we rerun that analysis. And when you do this, an interesting thing happens. Those fossils that kind of went somewhere near the base of the tree, they now fly inside a living group of daddy-longlegs, right? Now, why is that important? The reason that matters is that some of these fossils are among the oldest known exemplars of terrestrialization in the arachnids. That black and white fossil that you're seeing at the top, it's about 411 million years old. And what we're suggesting is that this is actually a member of a living suborder. What does that do to evolutionary inference? It turns out that it pushes back our estimated dates of when these animals colonized land. You end up with something like a 75 million year depth that is gained when you reposition these fossils. So here's a very odd case study where we started with a developmental biology question, but we ended up completely changing our understanding of a fossil, of evolutionary relationships, and of terrestrialization itself. So this then is our new understanding, and we're continuing to follow up by going after different members of that of the retinal determination network to understand the evolution of how those genes regulate one another. But this discovery of these vestigial eyes very much changed our understanding of the evolutionary biology of daddy-longlegs, and more generally, it points to the importance of looking outside of spiders and outside of that very specific context of whole genome duplication for changing our understanding of the evolutionary biology of this group. So in the past few minutes, I hope I've shown you how it is that our understanding of genomes, of genetics, of phylogeny, and of fossils are all informed by an integration of these different disciplines, and how it's very radically transformed our understanding of this fascinating group of animals. And this series of works was made possible by a very large number of people in my lab, past and present, as well as institutional support at the University of Wisconsin-Madison and several funding agencies, including the National Science Foundation. Thank you so much again for your time, and I hope a spider puts a smile on your face today.